welcome everyone. Invivoscribe has been providing products for hemato-oncology research and diagnoses for over 28 years and is the proud sponsor of this webinar. The title of today's webinar is A Comprehensive Solution for AML Precision Diagnostics. We are pleased to announce our speaker today is our very own Dr. Ying Huang, Senior Director of Product Development. She presented at PMWC 2022 a few weeks ago and is kind enough to present again for us. Dr. Ying Huang has over 25 years of research and industry experience with a broad knowledge in technology and assay development. Ying is passionate about developing novel methods using innovative technologies to improve lives with precision diagnostics. Her current work is focused on assays for diagnosis, stratification, and risk-directed treatment strategies in hematologic disease. Prior to Invivoscribe, she worked at Illumina, ACEA Biosciences, Nanogen, and MD Anderson Cancer Center. In those roles, she developed a variety of methods ranging from isolating CTC using microfluidic devices to detecting biowarfare agents using electronic microarray. She is a co-author of more than 60 publications and abstracts and is a co-inventor on 9 patents. Ying received her Ph.D. in electrical engineering from Bangor University, UK. For any questions during the presentation, please submit them using the chat feature. We will try to answer your questions at the end of the presentation if we have time. We have muted everyone, so please keep yourself on mute to avoid any background noise. And with that, I will turn it over to Dr. Ying Huang.
Thank you, Kay, for the nice introduction. So it's my pleasure to represent the presentation at the PMWC. So my topic today is a comprehensive solution for AML precision diagnostics. So acute myeloid leukemia, or AML, is the most common leukemia type in adults. So just in U.S., every year there's over 20,000 new cases. And the disease is very aggressive, the outcome is very poor. So it's only the 5-year survival rate is only 30%. So there's an unmet need to have a new therapeutic to improve patients' outcome. So currently, FDA approved medications for AML and can categorize into 2 groups. So the first one, chemotherapy, that one has been started since the 1970s, so it's going on for a long time. So the standard therapeutic, the 7+3, it's like cytarabine and also daunorubicin, those 2 combination plus stem cell transplantation used to be the standard care for AML for a while. Until very recently, 2017, the first target therapy, FLT3 inhibitor, midostaurin, started get FDA approved. So for the target therapy can be categorized to different targets. So the first one, as I just mentioned, FLT3 inhibitor. In my talk later, I'll talk about how Invivoscribe contribute to approval of these inhibitors. And also IDH inhibitor and recently the BCL2 inhibitor, and also the H-dog pathway inhibitor. So this is getting more and more like target therapy in this AML treatment. So with this new treatment adding up, it makes this decision for physician is complicated. So how typical treatment for AML is started induction, and then after that there's a consolidation, and after that the maintenance. Those are 3 steps. Within each step, there's different choices. So how to apply the best treatment choice to the appropriate population is really rely on the test. So for example, before the induction, it would be better to characterize the patient samples, looking for is any presence of the target therapy, such as a FLT3. So if a FLT3 mutation present, then maybe the patient should have the FLT3 in addition to the chemotherapy. Then after induction, so there's a standard like trial right now. People looking at the treatment of the induction. So you can do MRD test to see whether the treatment can has a better outcome, and also you can discover new mutations. So that can apply to the helping the physician make a decision, during the consolidation, what kind of choice should apply. So same apply to after consolidation. So based on the testing, we can make a decision, say what kind of maintenance options are better for the patient. So in the later presentation, I'm going to present Invivoscribe's comprehensive the test for AML. So total 6 different kind of tests. So the first one is our Leukostrate CDX FLT3 mutation assay. So this assay is a PCR-based assay. So primer is labeled with a fluorescent label. So then the PCR product is detected on the ABI 3500. So looking at the size, so there's size between the wild type and the mutant type. There's size different. So this assay, FLT3 has 2 types of mutations. So our assay covers both mutation, ITD and TKD. The Leukostrate CDX FLT3 mutation assay started as a single-site assay. Then we kind of like did additional tests, make it now is offered like as a distributed kit. So the kit including the reagents and also the software to analyze the data. So currently, this CDX FLT3 assay has been approved for 3 different FLT3 inhibitors. The first one, as I mentioned earlier in my presentation, was the first AML target therapy approved by FDA. So working with Novartis, so this FLT3 inhibitor midostaurin was approved in 2017 in US by FDA. And the same year, also, we got approved from Swiss Medic and also the EU. Working with Astellas, their FLT3 inhibitor, gilteritinib, we were able to get this CDX approved in US by FDA in 2018. And also later on the next year, 2019, we got EU approved. Working with the Daiichi Sankyo, we got crizotinib approved for CDX in Japan in 2019. So here we demonstrate working with the 3 different pharma companies within the 3 years. We have approved the 3 different CDX for the FLT3 mutation. So this demonstrated Invivoscribe's CDX development capability as supported by our expertise in different areas assay design, assay development, quality system, regulatory support submission, and our GMP manufacturer capability. The second assay, AML assay for characterization, is more like a gene panel assay because of the AML is a very heterogeneous disease. So this particular NGS-based gene panel assay is designed according to NCCN and ELN guidance, so cover almost 200 AML-related genes. So the mutation type, including SNVs, indels, inversions, and translocations. So the assay has been validated for CAP and CLEAR, offered at LabPMM. LabPMM is subdivision of Invivoscribe. So the average depth coverage for this assay is over 1,000x, and the LD is at a 5% allele frequency. Here I show some analytical and clinical validation performance for this MyAML gene panel assay. The top one is analytical. You can see the sensitivity and the specificity for SNV and indels, all about 95%. For clinical sample, with that many sample we tested, we have achieved 100% agreement, especially for those pathogenic mutations, FLT3, IDH1, IDH2, KRAS TP53. So after characterization, we would like to see the response after induction. So that will require a more sensitive assay. So that's why we have a MyAML assay. Unlike MyAML, this is more focused on these 23 genes. And the 23 genes is only look at hotspot genes. So with this assay, the average depth coverage is getting more, so it's almost 2,000x. And this assay also is validated for CAP and CLEAR. So here's some performance summary. So this LD has been pushed 1 log below the MyAML. So right now it's at 0.5% sensitivity. We also did linearity, LB, specificity, and also, of course, the clinical concordance. So we have over 90% agreement for the clinical validation. So talk about the multiple genes. We also offer a single gene MRD assay. So for FLT3 ITD and NPM1, so those two mutations are most present mutation for ML. FLT3 ITD is about like 25% AML patient has that mutation, and NPM1 is 30%. So we look at those two most present mutations and develop the single gene MRD assays. So here shows the linearity and LD validation data. So this single gene MRD assay pushes the LD even lower. So right now it's 2 log below the MRD, so at 5 × 10⁻⁵. For the FLT3 ITD MRD assay, we have demonstrated there's a relationship between MRD status versus the survivor in this populations in this gilteritinib study. So there's strong correlation using the 10 to minus 4 as a cutoff. So besides the MRD using molecular assays, most recently we also Flow Lab also developed the AML MRD assay using this BD lyric instrument. So this assay has 12 color and total target 21 unique biomarkers divided into 4 different tubes. Using the different from normal approach, this assay is very sensitive and can go down to LD at 10 to minus 4. The assay also being validated for clear and CAP. So the sample requirement being validated for bone marrow can either in EDTA or heparin. And the turnaround time is 72 hours from draw. And also the also has we also looking at the CD45 events. There's a cutoff for CD45 events. So here's some AML MRD flow assay performance. So the team did a method comparison, has a good correlation over 0.9997, and also the very good CV percent for the inter-assay, inter-instrument, or precision. And also the team also look at the carryover. The sensitivity of this assay is at 10 to minus 4. Also the LOQ is also at a similar range, 0.001%. So after we developed all these assays for ML, so being able to give more, people have more access to our assays, Invivoscribe also build infrastructure for the global lab network. Here shows we have a lab PMM in San Diego, in Germany, in Japan, and also there's lab, Invivoscribe also has a lab in Shanghai. So basically we have a lab across 4 countries in 3 different continents. So each lab offer at least different AML tests with the San Diego lab offer almost all the assays I just described. In addition to offer the test, each lab also support local clinical trial activities and submissions. So taking everything together, we can see Invivoscribe has developed a comprehensive solution for AML precision diagnostic through 3 different blocks. So we are using the MyAML gene panel and the Leukostract CDX FLT3 mutation assay for characterizations. We have MyMRD gene panel and FLT3 ITD and NPM1 MRD single gene assays, and plus multi-flow parameter AML MRD assays for MRD evaluation, and also our infrastructure global standardized network across the globally. So combining all these 3 blocks together, we offer a comprehensive solution for ML Diagnostics. So before I ending my presentation, I would like to give some introduction about our company. So our company was founded by Dr. Jeff Meller in 1995. So through over 27 years, the company has been focused on develop and offer global standardized assays by either kit to be sold globally to different labs, or by service to be provided by LabPM Global Network. So right now we have our product reach to over 700 different labs across over 160 countries.
We want to thank everyone for your attendance today at this webinar. Please note that a recording of this webinar will be available on demand on our website. We would especially like to thank Dr. Ying Huang for her excellent presentation. Thank you very much.